So what I'm going to talk to you today is our approach to congenital heart disease, which I hope to prove to you is fundamentally different in the way most people think about disease in general. And hopefully I'll convince you that it's the right way to go about solving this problem. What cardiologists and cardiac surgeons do is basically plumbing. That's all there is to it. The heart has 4 chambers. Blue blood, in which the oxygen has been sucked out by the body, comes back to the right side of the heart, goes to the right atrium, goes to the right ventricle, gets pumped out to the lungs. The lungs put oxygen into the blood, makes the blood red. Blood returns via veins to the left atrium, goes to the left ventricle, gets pumped out the aorta, where branches feed blood to all the parts of the body. Any rearrangement of the plumbing that you can imagine has been described in human beings. So veins may not be there or may not connect to the right place. Same with the arteries. Valves may not form or be severely misshapen. Whole chambers of the heart may be completely missing. The arteries might be switched. The ventricles might be switched. Totally bizarre things have been seen. They've all been described. So what we do is when there is a connection that should not be there, we close it. If there's a connection that's not there, but should be there, we make it. That's all there is to it. And I'll show you some examples of this. I told you that the blue blood stays on the right side, the red blood stays on the left side. This child has something called atrial septal defect, where there's a hole in the wall between the left atrium and the right atrium, so oxygenated blood can flow across into the right atrium, and this goes round and round between the heart and the lungs. This defect doesn't cause problems in children in general, but gone undiagnosed, it can cause really terrible problems in adults. So one of the things I try to beat into the pediatric residents is that no child should graduate from their practice with this type of defect. And so I teach them the nuances of diagnosing this particular defect. It's actually hard to diagnose. So when we see this hole in the cardiology clinic, what we do is we close it with a clamshell-like device. This device looks like 2 umbrellas. You have these fancy umbrella drinks. You can imagine both umbrellas going together. You can fold it up into a really small little thing and feed it through a catheter the size of a thick spaghetti noodle and deploy it so that the edges of this double umbrella grip the edges of the hole and close this defect off. Here's an example of where a connection is almost not there, aortic coarctation. So the aorta feeds fresh blood to the body. Sometimes it's pinched off or completely closed off after the blood is sent to the left arm. So what happens is if a baby has a completely closed off vessel, the babies typically will die because no blood is flowing to their vital organs below. And so in this particular case, this person has a more mild presentation of aortic coarctation. The cardiologist puts a catheter in, injects some dye that we can see on a video x-ray. You can see that the narrowing is right here. The next thing he does is puts a balloon in. This balloon looks like a sausage, blows it up, pops open that narrowing, and then we put in a stent, which looks like chicken coop wire to keep the vessel propped open. In this particular case, this is an example of hypoplastic left heart syndrome. So it's one of the worst defects that we see. It's hard to, these are all echocardiogram pictures done with ultrasound, of right atrium, right ventricle. And what you see here is an itty-bitty left ventricle. The left ventricle is so small that it cannot pump blood to the body. And so what we have to do is a series of operations, basically to make the right ventricle pump blood to the body and the lungs. Right? You need blood going to both places. Really, that's what we can do. But I think we can do better. So here's where it becomes a bit heretical. Biomedical research, the vast majority of biomedical research focuses on the causes of disease. The logic is that if you understood what causes bad things to happen, maybe you could develop a pill to treat the cause directly and prevent disease from happening or cure it. Right? The fact is, Scientists, doctors, epidemiologists have studied congenital heart disease for going on 4 decades now. And I can tell you, of all the factors, either environmental or genetic, that have been discovered, no plausible solution has been posed to change how we approach congenital heart disease. If we continue studying heart disease in this way, it's still going to be plummeting. The reason why is that none of the genes that have been discovered so far are amenable to drug targeting. So I won't go into the biochemistry and cell biology of that. You just have to take my word for it, that we are never going to develop a drug for any of the genes that have been discovered. So what we need is a plan B. Then where's plan B going to come from? So I think it comes from just thinking about the patients we see. This is a family that I saw. This baby here died as a toddler from hypoplastic left heart syndrome. And we thought nothing of it because the presentation of heart disease is generally sporadic. That is, we see a patient, we don't understand why the baby got a heart defect, we don't see any other family members who has a heart defect. When you do epidemiologic studies, the incidence of heart disease is about 1% in the population. But as soon as you find one relative who has a heart defect, the probability of finding another relative goes up about 5 times. And that's what happened here. A few years later, her aunt showed up. She was a teenager with exercise intolerance. So she was a varsity athlete, but couldn't keep up anymore. And we discovered that was because she had an ASD. We fixed it, and now I think she's graduated from college. She did varsity lawn hockey, field hockey. So that was a success story. Now, if you believe, as Dr. Schwartz says, that congenital heart disease is caused by a genetic mutation somewhere, we may not know the genetic mutation, but caused by genetic mutations, that suggests that this family carries a bad gene. So these 2 people got disease. But really, what's more interesting, if you're trying to develop a plan B, is to figure out why all these other people, the mothers, the fathers, the brothers and sisters, did not develop a heart defect, especially this person here, the father of this baby and the brother of this girl. If you could figure out why he didn't get a heart defect, maybe we could develop a pill that mimicked the activity of those good genes and give it to everyone else to prevent heart disease. So that's the logic. There are 2 possibilities. The other possibility is that he was completely lucky. He got the bad gene, but just lucked out and didn't get disease. And so what we can do are experiments to figure this out. Are there factors involved? If so, what are they, and how are we going to go out finding them? And the way I do this experiment is in mice. What I have is a mouse that has a genetic mutation of a gene, NKX25. It was the first gene discovered to cause heart defects in humans. This gene, this mutation sits within an inbred strain. We'll call it the black mouse. What an inbred strain is that you get 2 copies of a gene, one from your mother, one from your father. Within an inbred strain, each of those genes is identical. There are about 3 billion bases that encode all the genes in your body. A base is like a letter, and there are 4 letters in this genetic alphabet. And so every single letter is identical in when the mother and father give it to the baby. This leads to some interesting properties. So every father is identical to every son, every mother is identical to every daughter. The siblings, so all the mice within an inbred strain, are basically identical twins of each other. But then between strains, they're different. So within this inbred strain, when I knock out this gene mutated, there's a high incidence of heart defects. When I cross it to a white mouse strain, these babies, the first generation, are genetically half black and half white. Basically, none of these mice has a heart defect.